Yes. So you're going to put a yes there. Next column says chemical digestion. What does that mean in English, to chemically digest? Enzymes, you're melting, your hydrolysis, whatever word you like. You're melting the food, dissolving the food using enzymatic hydrolysis, whatever word you like. So, does your mouth chemically digest? Yes, that's called spit, right? That's why you have it. So I physically and chemically digest my cheeseburger. I have teeth and I have spit. Now, last column with yeses and nos. Nutrient absorption. What does that mean in English? Absorption? Got that. What does that mean in English, though? Absorb from where? Into your bloodstream. Very good. So I'm taking the food out of my organ and putting it into my bloodstream. Does your mouth absorb cheeseburgers? No. But Mr. Christie, what about nitroglycerin? We're not talking about pharmaceuticals. We're not talking about under the tongue, sugar pills. We're talking about like food. Put a cheeseburger under your tongue. It's still there. I've tried this. I have a friend. Sorry. All right, so food we do not absorb in your mouth. Right? So we're talking about food. So you're going to have a yes, yes, and a no. When you get to the pH column, I don't need a number. I just need a value. Is it acidic, neutral, or basic? Okay? I'm not going to tell you that one. When you get to enzymes present, that's not a yes or no. I need the names of the enzymes in the mouth. Your lab manual has a sheet, so does your book, and so does your phone. So you can do that. Anyone know what enzyme source means from Biology 112? Where they're made or where they're produced. So they're in my mouth, but where are they made? This wording comes from 112, that class none of you should have passed. Right. Let's review Biology 112. I think it's the fourth lab of the term. You had something look like this. You had an E, which was an enzyme. You had a little triangle that went into it. Right? You drew something like this. And then the next week you did something kind of like this. Right? The S represented substrate. So in English, that's what the enzyme works on. So substrate is what you started with. What did you call the two little things that you made over here? Products is what you made. So for each enzyme that you list, you're going to list what goes in, what comes out. Substrates and the product for each enzyme. Make sense? Okay, your job, I'm going to give you about 10-15 minutes with your team, divide and conquer, but fill that in, we'll go over it. Have fun. Into my blood, right? So let's do two together. Give me some symptoms if you go way up high. What else?
Increased respiratory
rate, nausea, headache,
increased heart rate,
blah, 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 all those things, right? So go into Denver tonight, tell me how you feel. You have headaches, all that stuff. Because my body's getting less oxygen to the tissues, right? So now we got to do this number 3. What would my body do up there? So we're going to learn something. You're going to learn hypoxic drive. So, medical terminology people, tell me what hypoxic means, or hypoxia.
not enough oxygen.
Low oxygen. So, I'm going to define it for you. So if the oxygen levels in your body are really low, how low is really low? Yeah, so you got about half-ish, they usually say. Don't quote that number. So if your oxygen levels drop by about half, what will happen to you if you run out of oxygen? You're dead. So if I were a brain, and I realized that I'm running out of oxygen, what would I tell your body to do? I'm going to breathe faster. You're going to start hyperventilating. Because the brain thinks the reason you're running out of air is you're just not breathing fast enough. So if oxygen levels get really low, you're going to start hyperventilating. So let's go back to your notes from last week. Last week I screamed and yelled, what drives breathing rate?
CO2.
CO2, because you're at sea level. So at sea level, oxygen's always high, right? There's no reason to worry about it. So under normal conditions, acid drives breathing. If you go way up on top of Mount Denali, this becomes a problem. So your brain has a backup system called hypoxic drive to accommodate the fact that you're idiotic enough to go up all that way. Let me show you a picture. This is showing your brain. And remember, these are the respiratory cylinders you learned last term, right? Review for me again. What's this big guy here? Pons, this one here. Medulla, remember both of them did breathing. So if you look, everything has CO2 and H. CO2 and H. That's the acid level that we talked about. But notice, there's also this one. That's the hypoxic drive. That's a backup system for just such an emergency. So if you go on top of Mount Hood, this becomes low enough that your brain starts realizing I need to change your breathing rate or you'll be dead in 2 minutes. Make sense? So on a test, unless I say high altitude or something, you're not going to assume hypoxic drive. It's only when oxygen is really, really low, which is abnormal for most human beings, most of the time. Make sense? But you have this system called hypoxic drive. Let me show you that I'm not making this up much.
I'm so sorry.
Let me show you this picture, a little animation here. This is actually a whole website on high altitude stuff. Let's look at this. To show you I'm not making this up. Here's some math stuff for you. They're doing it by height, and no, I'm never going to ask you to test about these numbers. That gives you an idea. So as you go up in height, the amount of oxygen you get is decreasing. So let's go down here. All right? So at 3,000 meters, your oxygen basically is 50% of what you expect at sea level. If you go past that, then it drops way, way down, right? So you're dropping below 50% as you climb higher and higher, especially over 6,000 meters or so, right? So that creates this problem where your brain realizes I'm going to be dead unless I speed up your breathing. Make sense? So it's an abnormal sort of hypoxic drive system. So let's go back to our case. So, what happens in number 3? When you lose all that air, what happens to your hyperventilation? More or less. You're going to hyperventilate more. That's hypoxic drive. So then that leads us to question number 4. What happens to your blood gases, say ye, if I breathe faster? Tell me what oxygen should do. So, should go up, assuming I have some in the air. CO2 should go down. Right? Because I'm exhaling more often. So if I start breathing faster, my oxygen level should climb, and my CO2 should go down, right? So I'm inhaling and exhaling. So then let's go to question 5. That's that stinking, yes.
So do lungs not have quite the same problem as the heart, where if you start beating faster past a certain point, it doesn't, I mean, I know gases can move a lot more quickly than a liquid, but do you start having decreased yield or decreased exchange once you get past a certain
Actually,
rate?
it's sort of the same thing. There's only so fast you can expand a lung. And so what happens is you can only breathe so fast, and then at some point you can't keep moving a lung like a heart. You have to move your whole rib cage. You can't move your rib cage that fast, so eventually there's a limit. Okay. Right. So there's a backup system for that. So let's go to number 5. That's that hemoglobin curve I just erased over here. So let's draw it again and see what will happen if I breathe too fast. Percent RBC. Right? Remember that curve? Right? So looking at the blood gases over there, tell me what my curve will do over here. Will it go up or down? Or right or left? Here, here. Here? Here? Here. There. If you think about it, it's going to go that way, right? So let's review. Things that make it go to the right are things like more acid and more temperature. I have less acid in my body because I just blew it all away. So my curve will shift to the left or up. Make sense? Tell me what that means in terms of unloading for oxygen. Less unloading, right? Bingo. So if I'm on top of Mount Hood, I'm giving less to my tissues. Is that a good day? No, because I have less anyway, and I'm giving less to my body. So my body just did the reverse of what you want because I just purged all the acid out over here. Makes sense? But the brain doesn't know you're on Mount Hood. It thinks you're at sea level. So luckily for you, your body has a backup plan. Go down to question 6. There's a chemical that your body makes called 2,3-DPG. Let me show you a picture of what 2,3-DPG does. Egad, it's a curve question. Look what happens as I increase the 2,3-DPG. What is my curve doing? Which way is it going? To the right. Let's draw that again and see if we understand. So if I do DPG, bless you, I shift rightwards. Tell me what rightwards does for my unloading. I get more unloading. So by making this chemical, your body shifts your graph more to normal. You're unloading more again, which is what you want at altitude. Make sense? So if you know people who climb, what are they supposed to do every day or every couple of days? They're supposed to do what?
Acclimate.
They stay there, right? Base camp idea. So think what they're doing. They're going up in altitude. They're giving their body time to make more what? More of this. Therefore, I can keep shifting my curve rightwards to unload more and more oxygen to my tissues. Yes?
How is this related to EPO?
Hold that thought, because we'll get to EPO soon. So if we go back to our case, it takes a day or so for your body to make this chemical. So the first day you go to Denver, you're going to be miserable. By the end of the weekend, you'll start making DPG and shifting your curve back to normal. Make sense? So that's how your body's compensating for the hyperventilation from your brain. Fair enough? So you shift right. Yes?
So if you live somewhere like Denver, you're just making that all the time?
Yes,
So that you can...
to a point. And then we have this question to answer this question. Let's go to our study. We have Mark, who's going to die. Not really. And we have Emily, the RN, who knows her biology. And look what she says. We spent a month at 15,000 feet. So go back to last term. When you live in Denver, what changes in your body?
Your blood.
Your blood. How does it do that? Remind me for question 8 how I change my blood.
You make more.
Let's see. Less oxygen makes what? More EPO, which makes what? More RBCs, which do what? Carry oxygen in my blood.
Isn't that called the RAS with 2 A's?
What? Oh. So if you remember 232, if I go there over weeks, I'm going to make more RBCs to carry more oxygen in my blood. That's a long-term strategy, right? The thing we just did is a short-term. So DPG is days. This is month. So if you go to Denver for the weekend, you'll make DPG. You won't make more EPO. You live in Denver for a month, you'll make EPO. That should be reviewed from 232. So if you go to Denver or you live in the Himalayas or wherever, you will have more blood, and that will help compensate for this problem here. Make sense? That's a long-term strategy. So let's figure out what's going to happen to Mark. He keeps going downhill, right? Figuratively, right? Emily puts him on life flight. Good job, Emily. And the EMTs give him oxygen in the Gemo bag. So, why would you give him oxygen? Gee, aren't we trying to get oxygen to go into his body, right? So we're doing diffusion. What about the Gemo bag? Why put him in basically a pressure cooker?
To make more RBCs.
Not so much the RBCs.
Help with diffusion?
Help with diffusion, right? If I increase the pressure and the amount of oxygen, I should get more into my body. So we're trying to increase the oxygen diffusion into him. Give him oxygen and basically force it into his lungs. Make sense? Right. So here are you going to do, future nurses that you are. Here's some data taken from Mark. We're going to go through this as a class. Let's figure out where these things are. So arterial oxygen, where is that in Mark's body? In his blood, right? Okay, Mark has 52. Normal people have about 100. What's your conclusion? Pretty lousy, right? He has low oxygen in his blood. Okay. CO2 is 30 in Mark. It should be 35 to 45. Conclusion? Low. Hey, it's kind of partly this, right? I understand that. Okay. Now we have these other ones, which are chemical stuff. I'm just going to tell you those are normal. Those have to do with the chemistry of it. But look at alveolar oxygen. Where's alveolar oxygen? It's high, but where is that?
In your lungs?
In your lungs. So it's actually high, right? Which makes sense, they're giving him oxygen, right? And this is considered normal. So now we're going to go down here, which lab result is the big problem? So of the three things that are odd, the first one, the second one, and this one, which one's the most critical to Mark's well-being?
Arterial.
Arterial what?
Oxygen.
Oxygen. And why is that life-threatening?
Slow.
Right. You have half the oxygen, you have half the lifespan, right? You're going to die. So we have to fix his oxygen levels, because otherwise you're pretty much going to die, right? 52 is not good. So now we got to figure out why it's 52. Look at his alveoli. He has high oxygen in his lungs, and he has low oxygen in his blood. So can you tell me where the problem is? And what do we call that little place between blood and lung? Respiratory membrane. So he has a problem getting the gases to leave his air sac and get into his blood, because his air sac is high, his blood is low. That has to be a respiratory membrane. So how could you screw up a respiratory membrane? You could be a smoker. Let's blame him on smoking. What else could happen to your respiratory membranes climbing in the cold, dry mountain? Become scarred, they can become dry, right? They can become damaged from the cold. So all those things are affecting his air sacs, and we're getting less efficient at getting oxygen from the air into his blood. Which has a problem with his respiratory membrane. It's actually pulmonary edema, formally. Make sense? Mm-hmm. Right. So now we got the harder question, the very last question, number 12. 7.23. What is that in terms of people? That's low. But wait a minute. CO2 is low. If CO2 makes acid, the pH shouldn't be low. So why is this pH low if the CO2 is actually low?
He's still not blowing it off properly.
Well, he is.
Is his temperature low?
No.
He's blowing it off too much to compensate?
No. I'm never going to ask you something this hard. Okay. Go back to biology number 12. Why do cells need oxygen?
To produce energy.
And what's the byproduct when you produce energy with oxygen? You make what? CO2. But now I don't have any oxygen for my cells to make energy, right? So what do cells do if they don't have oxygen?
Anaerobic.
They do anaerobic. What's the end product of anaerobic respiration? Say it again.
Lactic
What?
acid.
So the more I run out of oxygen, the more anaerobic I do, which makes me more acidic.
Isn't it technically fermentation, not anaerobic respiration?
No, in humans actually it's the other way. You have to say anaerobic. We don't ferment, because we don't make alcohol.
I don't know, in microbiology, they were very insistent that only prokaryotes undergo anaerobic respiration.
And they'd be wrong, because yeast do fermentation, they're not prokaryotes. I would disagree with whatever your instructor was. Right. But let's go through it. So in English, they're getting acidic, but it's not the CO2. This is acidic from other mechanisms, from anaerobic. So I'm never going to ask you to test to claim that you know that. So what you're supposed to understand from this case study is actually the first page on that curve. So my curve went left when I lost my acid. The chemical shifted it right to get the unloading back to normal. You understood that concept. You understood how this is all playing out. Make sense? Yes?
So if you have pulmonary edema, do you have to remove that water?
Yes. Either you evacuate it, or you just hope he pees a lot, or you get his pressure back up. So one of the things that cause the edema is the pressure drops, and therefore the fluids just leak out. Yeah. Make sense? So if you understood the curves, that was the point of this whole exercise.
I don't understand how he got pulmonary edema.
Whenever you have low pressure on the outside of your lungs, there's nothing to keep the blood from punching out. So as the pressure outside goes down, the force of the heart overwhelms the capillary wall and blows the fluid into whatever space they can find.
Oh.
Yes.
So that's been happening his whole
Yes.
Oh, okay.
Or we can call him a smoker.
Unfortunate.
You okay with that? That's a curve idea. So thankfully, you're now going to leave respiratory system entirely behind. So we're all done with All right, for mouth, we had yes, yes, no, which I told you. pH. Did anyone look up a pH for me? 6.7, which we're going to call neutral, just to make it easy on you. All right, are there enzymes in your mouth? Yes, they are. Someone name an enzyme I find in my mouth.
Salivary amylase.
Salivary amylase. All right. So now you have to go back to Biology 112, that land you forgot, and figure out what the coding is. So in enzymes, the rule is ACE means enzyme, right? This thing here means something, and this word here means something. The prefix is the substrate, or the enzyme. So does anyone know what amyl is in biology? That's starch. So amylase breaks down starch. So we have amylase is the enzyme. The substrate is starch. I have to go back to 112. What does starch become when you digest it? Maltose, monosaccharides. We'll just say sugars and call it good for today. Because there's actually diet. So we're going to say starch into sugars. Okay? So over on product, I'd say sugar. So the substrate is starch, the effect is sugars, yes?
Can you pull the camera down?
Well, if I can do that, then
Yeah, sorry. There's just a-
yeah, no, I can't write on the screen. I did want to. I'll do that. All right, so let's try this next one. The source. Where do you think salivary amylase is made? That's what this word is. Traditionally, the word in front is the source location. So if you remember how to name the whole thing, you have three things: enzyme, substrate, and source. Salivary amylase. So it's made in my salivary glands. So amylase is the enzyme, salivary glands is where it's made, converts starch to sugar. So far so good? Name another enzyme in your mouth. Lingual lipase. I love hearing two names. Lingual lipase or lipase. Lipase means enzyme. So I'll do lipase here. Can someone guess where lingual lipase is made? On my tongue, because that's what lingua means. So the source is your tongue. Okay, it digests lips. So what is lip in biology? Fat. So in biology 112, fat or lipids become what? What's the monomer of fat? Fatty acids. Very good. You can argue glycerol as well. Let's do that. So lipase is going to digest fat into fatty acids. Make sense? So that's our enzyme for our mouth. Okay, mouth? So let's try the esophagus. Tell me, does your esophagus do physical digestion?
No.
No. It pushes food, that's not making food smaller. So no. Does your esophagus do chemical digestion? No, it does not. Does it absorb anything? No. What pH is it? Same as the mouth. What enzymes are there? None. We hope that's the only one on the exam. Right? Nothing, nothing, none. Because all it does is get food to the stomach. Stomach. Does your stomach physically digest?
Yes.
Yes. That's what your stomach's doing when it's growling, churning, rumbling. It's like a cement mixer. Does your stomach chemically digest?
Yes.
Yes, it does. Vomit and you'll see.
Oh my goodness.
Does your stomach absorb nutrients?
No.
No. There are some things it can absorb, like alcohol, aspirin, whatever, but in terms of food, it doesn't absorb food products. That's why your vomit contains your dinner. Right? pH. Give me a stomach's acidity level.
Acidic.
Really, really acidic. pH of 2 or so. Now let's do the enzyme. Give me some enzymes in your stomach. Pepsin. All right. Here's a problem. That does not say ACE, and there's a word missing. You just got to go with it. Some of the enzymes are named long before they came up with the system. So this is still an enzyme, even though it's an N, and even though it's missing the word here. So let's figure out what pepsin, where it's made. Can someone tell me where pepsin is made? Chief cells of my stomach. Peptic ulcer, Pepsid-AC, Pepto-Bismol. They're all telling you pep, pep, pep. All right? That also tells you what the substrate is. What does pep relate to? Peptides, which is a fancy way to say what? Protein. So, pepsin digests protein as a substrate. From Biology 112, what's the monomers of proteins? Amino acids. You can say oligopeptides, that's fine. We can go AAs. Now break down proteins into amino acids. You okay with that? Give me another enzyme in my stomach. Gastric lipase. Oh, I know that one. So lipase breaks down what to what?
Lipid.
Fat, same as the other one. They're the same enzyme. So fat to fatty acids. Difference is, where do you think gastric lipase is made? In the stomach. Same enzyme, different source. So it'll also be from your stomach. You okay on those? All right, let's add in that last column. There's some other stuff in there. Name something else you'd find inside the stomach. Hydrochloric acid. So you have the acid or HCl. Anything else you might find in the stomach? That's the precursor to that one. Oh, so help you in your homework. There's something called intrinsic factor, which I ran out of lines. This helps you absorb vitamin B12. This is not an enzyme in the sense of digesting food. It helps you absorb vitamin B12. So it is like a cofactor. But that's not an enzyme for digestion that's found in your stomach. Okay? The only two enzymes for food are these two that we're talking about. Make sense? Okay, let's go to small intestine land. Does your small intestine physically digest food? Say yes.
Yes.
Yes. Now let me show you what we mean by that. So before you yell at me, let me show you a picture. Where'd it go? Add it up here. There we go. There we go. So this is showing what your tubing can do. So this is what your esophagus is doing. It's just pushing the food, so the green is your Whopper with cheese, pushing the food down your stomach. Notice it's the same size and shape and color. Nothing has changed, so I'm just pushing food. Fancy word peristalsis. This is your small intestine. So to make the green the Whopper with cheese, there's my fries up there. So I'm taking half the Whopper, half the fries, mixing those together. So the food's actually getting smaller physically in my small intestine. Fancy what's called segmentation. But that is considered a digestion. This is not. Right? So pushing is not digesting, mixing is. So the small intestine mixes, therefore is considered a yes. Make sense? Mm-hmm. All right, so we have a yes for our small intestine. Go back to the big screen. So does your small intestine chemically absorb? Yes, yes, yes. Yeah, we'll put a little star there. We like it. Does your small intestine absorb nutrients?
Yes.
Yes! Put a big star and a couple things there. Yes, yes, and yes. Here's the question. Why is your small intestine absorbing and not the other ones? What makes the small intestine different from the other organs? Let's find out. Let me show you a small intestine up close and personal. Right? Where did I lose it? I lost my small intestine. Still alive.
I hear those are pretty long. That'd be hard to lose.
Yeah, they are pretty long. All right. Here's a picture showing some small intestine. Notice it has these folds in it. What do you see on the tip of those folds?
Cilia.
Actually called villi fingers.
Oh,
but
villi.
you got the same idea. So you learned in second grade there's villi in your intestine. Okay. Here is a villus. What do you see? What's that red and blue stuff in there?
Capillaries.
Capillaries. Tell me from last week what the job of a capillary is.
Division.
Exchange, right? All that. So when food hits my villi, it can enter my bloodstream and be exchanged. So I'm going to absorb food in the villi of my small intestine. If you look closely, the villi have fuzz. Here's the fuzz of my villi. So I have fuzzy fingers with folds. This is really what your small intestine has. Here's the microscope of it, right there. So from 231, do you remember what type of lining is in your intestine?
Pseudostratified ciliated columnar
Not
epithelium.
stratified.
Well,
Not even pseudostratified.
Columnar epithelium.
Simple. Remember, it was simple. Why? What was the job of simple tissues?
diffusion.
Absorb and secrete. So if you think about what it says, how many cells are between my cheeseburger and my blood? One cell. Just like in your lungs, it's easier to diffuse through a simple thin layer. So your small intestines are very thin. They have one layer, and they have a lot of surface area, so it makes it very, very easy to absorb. The other organs are not so neat. I'm going to pick an esophagus just because I can.
So does the small intestines absorb certain things than others?
Absorbs pretty much anything that you get in.
Okay.
So there's only, I think, I can't even think of the exception. It absorbs pretty much everything.
Okay.
So on this microscope slide, this is your esophagus. Remember your esophagus? What kind of layer did it have?
Ooh.
Stratified. Why did it have stratified? Protect. That's the Dorito effect, right? How easy can you diffuse through multiple layers? You can. So one of the tricks is my small intestine is much thinner, has much more surface area that gives me a chance to absorb much easier. Same idea as your lung. Your lung and your intestines basically share the same kind of concept. Very thin, very big surface area. Make sense?
Mm-hmm.
Your small intestines where you do all the absorption, mostly. Make sense? All right, so let's go back to our chart. Now we have to do the enzymes of the small, pH. The pH. Go to the pH of your small intestine. It's basic.
Why?
We'll get there. Hold that thought. Well, I said hopefully by the end of the night.
Maybe it doesn't.
Okay, now we have to do the enzymes. Clear a big page. Because thou shalt know every enzyme, PCC says, of the small intestine, PCC says. And when my pip talks, I listen. All right. So, you are supposed to know every enzyme in the small intestine, which I think is about 20 some odd.
Oh my
What I've found works easier if you break it into categories. Learn the ones in each group. It seems to work easier. So what we're going to do, we're going to first do the enzymes that deal with carbohydrates. And we'll list them all out, and then we'll go down the road. So, can someone find for me on a list somewhere a carbohydrate enzyme of the small intestine?
Amylase.
What kind of amylase?
Pancreatic.
Very good. I know, these will not all fit in the box. So let's review. ACE means enzyme. What did amylase break down? Starch into what? Sugars. What is the source, say ye? The pancreas is the source. So pancreatic amylase is made in my pancreas, but it goes to my small intestine to break down starch. Same amylase as in your mouth, just different location. Make sense? All right, let's name another carbohydrate type enzyme. What is it? Ooh, I'll change the wording on that one. I'll make it the easier, older word that I like better. You have maltase, lactase, and sucrase. Dextrinase is basically in here. Right?
There's also glucose.
Yeah, we'll skip that one for today. We'll make it the easy wording. So here's maltase, lactase, and sucrase. Let's review. Can someone tell me what maltase digests? Beer. Yes, indeed. Malt sugar. So this is maltose, which is beer sugar, oats, barley, anything with grain. And what do I make when I break down beer? Sugar, glucose, right? That's why you get fat when you drink. All right. Lactase. This should be easy. What does lactase digest?
Milk.
Lactose is the sugar. And what do I make when I break down milk? Glucose again. Hey, I'm catching on. So tell me, sucrase would digest what? Sucrose, table sugar, into glucose. So all three of these are breaking down some, not complex sugars, but disaccharides into monosaccharides. The problem is they didn't put the first word on. So where's the source for these three enzymes?
they all come from the pancreas?
They do not, actually. Nope. Nope. Well, yeah, but not. I want more of an organ. Oh! If you read Saladin, he will hit you with this word. These are brush border enzymes. So the problem is, where in the world is a brushy border? So, if you're a drunk Roman, brush border means it's a fuzzy edge. Can someone name an organ that we just talked about has fuzzy edges? Small intestine. So when you see the word brush border, all that means is your small intestine, because it's brushy on the edge to some drunk Roman. So a brush border is synonymous with the small intestinal lining. They're the same. This one just costs you more. So all three of these are brush border enzymes, meaning they're made in the small intestine for action in the small intestine. You okay with that? So you have one pancreatic enzyme and 3 brush border enzymes. You okay with our carbs? Mm-hmm. Okay, let's do some protein. I'll go right here. I'll use purple's good protein color. So let's do some protein. Can someone name a protein enzyme for me? Peptidase. Okay. There's also carboxypeptidase. There's trypsin. There's chymotrypsin. And a whole battery more, which we're going to ignore. So I'm going to make you learn these 4. All right, so all these ones are going to digest proteins. So what do you make when you digest the protein?
Acid.
What kind of acid?
Aminos.
So all these ones would make the same monomer at the end. They're going to take proteins into amino acids, just like your pepsin did. There's no real difference here. There's a technical difference now. The problem is you don't have a source word on these. So let's walk through it. We'll start at the bottom. Can someone tell me where chymotrypsin is made? You don't know. I might as well just say anything. So formally, it's a pancreatic enzyme. So it's made in my pancreas, given to my small intestine to digest proteins. Trypsin is also a pancreatic enzyme. Meaning it's made in my pancreas to my small intestine. All right, carboxypeptidase is a brush border enzyme and also a pancreatic enzyme. Oh goodness, can you do that? Can you have 2 sources? Sure you can. So that's one that's actually in 2 different places. One is the small intestine, one is the pancreatic. Peptidase, the one I'm talking about, is a brush border enzyme, meaning small intestine. There's actually different categories of peptidases. So some of them are made in small intestine, some are made in pancreas, one of them is made actually in both. You okay, our proteinaceous ones? Okay, we got carbs, protein. What's my next one going to be? Let's do fat. Fat's fun. Someone name for me a fat digesting enzyme. Lipase. What kind of lipase? Actually, you can see it right there. You wanted to say pancreatic lipase. So what did lipase digest? From what to what? Fats into fatty acids, right? No different than the other lipase. The difference is the source. Where do you find pancreatic lipase? Made in my pancreas for my small intestine. Very good. So pancreatic lipase is a pancreatic enzyme for my small intestine for fats. Now, let's do our last group. Our last group, if you remember biology 112, of macromolecules was nucleic acids. So, the enzymes are called nuclease. All right? So, digesting nucleic acids. Let's figure out what you make. When you digest nucleic acids, what do you form?
Nucleotides?
What was that? I heard it.
Nucleotides?
Nucleotides. Amen. Nucleotides are the monomers of nucleic acids. Does that ring a bell? Where do we make nucleases? More than one. I'll give you a hint. Pancreas is one.
Brush border.
And brush border is the other one. So there are more than one source for nucleases. So on a test, you really can't go wrong with pancreas. You don't go wrong with brush border once. But pretty much, you're supposed to know every category, the enzymes within them, their source, their products, their substrates, and all that. Make sense? So the small intestine is one that's going to eat up all your life. Make sense? Oh wait, we're not done. Because we got enzymes, sources, products. There's something else inside your small intestine that I need to put there. What else do I find in the small intestine?
Nutrients? No.
This would be bile.
Oh.
So we're going to go over here and talk about bile for a minute. So bile, anyone know where it's made? In my liver. Anyone know where you store it? Gallbladder. Alright, so liver makes it, the gallbladder is where you store the remaining. Okay, so now we've got to figure out the function. Can someone tell me what bile is for?
Break up emulsifying.
Very good. So you'll see different words. You'll see people say emulsify fats. That's legit. You can say it mixes fats and water. That's perfectly legit. You can say it helps blend fats. All those things are perfectly legit ways to say it. Right, let me show you what we mean by that concept. Let me show you a picture of some bile. There we go. So here's my triple waffle with cheese inside. And here comes my bile, the little guy on the side. So here's my bile. Oh look, biology 112 words again. So polar, right, hydrophilic, nonpolar, hydrophobic. But here's my bile, here's my whopper. And what did it just do here? What's it doing to my whopper? Binding to the fat and doing what with it? What's it look like it's doing to the fat? Yeah, it's making the smaller pieces. It's emulsifying, it's mixing, it's chopping up, however you want to phrase it. Right? So can you name something in your kitchen that you use to help break fat up? So, congratulations, you now know what bile is. Bile is dish soap. It's exactly the same chemistry. There's absolutely no difference between bile and dish soap.
I don't want to be done.
You add dish soap to break the grease off your dishes. Bile breaks the grease off your meal. It's the same logic. However, one thing to keep in mind, bile is not an enzyme. A classic exam question, I ask you enzymes, you put bile and you miss a point. Because this is not an enzyme. The soap doesn't digest the fat. What digests fat? Lipase. This is the enzyme. This digests fat. That just makes it smaller. Notice that I still have the same amount of fat here. Nothing has changed. All I'm doing is making it smaller bubbles. The lipase would make it vanish. If you pour soap on your sink, your greasy pizza, all you see is a bunch of bubbles of the fat. But the fat's still there. Okay? So an important concept is that just makes the pieces smaller for the lipase to work. Make sense? So that's a soap or emulsifier. It's not an enzyme. That's the enzyme. But that is in my small intestine. Make sense? So, good. So let's go back to our chart of doom. We're almost done. Large intestine. Tell me, does your large intestine physically digest? No, it pushes poop. That is not digestion. Does your large intestine chemically digest? No. This is poop. That is not digestion. Does your large intestine absorb nutrients? Yes, it does. Now we have to specify here. What nutrients does your large intestine absorb? Okay, we have water, vitamins, and salts. Anything that can be in the water, I could absorb. So things that are water-based. So water is a nutrient in this class. So let's go through it. pH. It's basic, although I guess people are turning. So now let's do the enzymes. Name the enzymes in your large intestine. Why are there none? Because it doesn't do digestion, right? So, nothing. Yes.
So you mentioned that you're going to tell us why it's basic rather than acidic.
Yes. Give me time. I'll get you there.
Darn it.
So okay in our large intestine? So you hope it's like the esophagus, just with food. Right. Let me show you an animation of this process. Blood esophagus. Sounds really good. See what McGraw-Hill has to say.
The primary functions of the digestive system are the breakdown of food, called digestion, and absorption of nutrients. Digestion begins in the mouth, where the teeth break food into smaller particles during mastication.
All right, so mastication is an uncertain joke, all right? But that's physical digestion. I'm just breaking the food out. Yes.
Salivary glands, located near the oral cavity, secrete saliva, which begins chemical digestion and keeps the food moist.
There's my spit. I'm trying to dissolve the food. So I'm yes and yes because I've chewed it and spat upon it.
As food is swallowed, the soft palate blocks the upper pharynx to prevent food from entering the nasal cavity.
So notice that the food is still there. Why is it still there? Because I didn't absorb it, right? It's still there. So I'm now going to pass that food down. Hey, what's that funny looking thing from lab? Epiglottis, right? So we want the food to go to the back tube, not the front tube.
And multiple voluntary muscles in the face, neck, and tongue contract, pushing food particles through the pharynx. The food passes over the epiglottis, which prevents food entry into the respiratory system, and then into the esophagus, which connects the pharynx to the stomach. The one-way movement of the food mass, now called a bolus, is controlled by wave-like involuntary muscle contractions. This movement is known as peristalsis.
But notice the food is still there, right? I haven't done anything to it. So that's why it's nose all the way across. I'm just pushing food. Say hello to Mr. Stomach.
The bolus now enters the stomach. Folds in the stomach wall, called rugae, allow for expansion as the stomach fills. Stomach cells secrete hydrochloric acid, pepsinogen, and various regulatory hormones that chemically digest the bolus.
So there's the chemical digestion, aka vomit. So I'm melting the food.
Muscular contractions in the stomach churn its contents to further break down the bolus and mix it with stomach secretions to form a thick liquid called chyme. Chyme exits the stomach through the pyloric sphincter and enters the small intestine, the major site of nutrient absorption. The small intestine consists of three parts: the duodenum, jejunum, and ileum.
Duodenum's fine.
Bile from the liver and digestive enzymes from the pancreas empty into the duodenum to aid in digestion.
All right, so here's the important part. The liver and pancreas don't actually touch the food. That's why they're not in your chart. Right? But they're helping the small intestine. The liver makes bile, which is my soap. The pancreas does all those enzymes there. So they're accessories. They're helping. But the food doesn't go through your pancreas. I hope not. That'd be bad. But they're giving liquids to the small intestine for it to better do its job. They're helpers.
Absorbed nutrients pass from the lumen of the small intestine into blood and lymph.
And why is it able to go into my blood there? Little villi, it's thin, all those things are there. Large, small surface area, all that stuff. Let's go to the colon.
Chyme not absorbed in the small intestine enters the large intestine. As it passes through the cecum and ascending, transverse, descending, and sigmoid colon, water and salt are absorbed, and chyme is converted into feces.
So we're absorbing the water out of the remaining food and the salts that go with it. Now we made food.
The rectum stores feces until nervous stimulation initiates the defecation reflex, resulting in elimination through the anal canal.
You're all hoping they show that, right? Yeah. I'm thankful they didn't. Right? But that's your chart basically in a nutshell. Right? You okay on the organs? All right, so now we're going to add a little detail to your notes. So go back to your pack of the pain and scroll down a couple pages to this case study. We're going to flesh out some details. Sure. Yes. Because it also affects all your insulin glucagon, and also you can't digest anything with lost pancreas. So you start to that. Pancreatic cancer is this. Right. The loss of insulin glucagon immediately makes your blood sugar rise. And then you can't digest anything, so you get jaundice, you immediately become malnourished, you shrivel up really fast, you're dead too much. Pancreatic failure. So let's go through this. So we'll do this kind of as a class part of it. So we got this business guy, you know, he's got heartburn, it's after he eats, he belches a lot, blah, 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 pays for expensive tests, and they say, congratulations, you have some kind of gastric reflux. All right, tell me in English what gastric reflux means.
Heartburn.
Acid from my stomach backed up. So if you look at your chart, one of the things I find in my stomach is acid. And that reflux means back up. So we used to call it heartburn, and it got expensive, right? This is GERD, gastroesophageal reflux disease. But now I have to figure out number one. Well, what should keep the acid out of my esophagus? I heard it. What was it? Sphincters. We're going to learn your sphincters. You think I am probably one, but that's okay. Right. Which one? That's the important question. So let's learn our. So PCC says, "Thou shalt know your sphincters, thou shalt." So we're going to go through sphincters. So in English, what is a sphincter? It's a valve. The valve with a slightly different anatomy. So go back to 232. Why did your heart have valves? Stop backflow. So we want to prevent backflow. You really don't want your poop coming back up, right? Last time I checked. What else did valves do for your circulatory system besides prevent backflow? They regulate the pressure. So another thing we can do with valves is we can regulate the flow. So your digestive system is going to have valves just like your circulatory system did to help control the flow. We call those sphincters. So you're supposed to know all the sphincters and where they are to answer question one. Let me show you a couple pictures, and we're going to walk through the body in order, as though I were a cheeseburger. Let's find some. So here's a picture showing our first sphincter of the night. And someone named the sphincter shown here, some big bold letters. Upper esophageal. So let's do our first one. Number one is the upper esophageal. In English, where is that? Back of the mouth. Or you can say between your mouth and your esophagus. You can say pharynx. I don't care how you say it. Just understand that's the one right here. Right? So, my upper esophageal is closed. Here comes my cheeseburger. I have to open the door. So I relax the sphincter. That way the food now can enter my esophagus. Right? So that's my upper esophageal valve or sphincter, to be formal. Make sense? So if there's an upper esophageal, take a guess what the second sphincter would be.
Lower.
The lower esophageal. So let's find that one. Unfortunately, this one has 12 different names, it seems like. But we're going to call it the lower esophageal, just to call it one. So number two. Lower esophageal. On this overhead, they're calling it the gastroesophageal. I learned it as the cardiac, the LES, the cardioesophageal. Pick a word, I don't care. Let's do this one. Where is that? How would you describe where it is? Top of the stomach. So if you think about your heart again, after every chamber was a valve, right? If you notice, after every organ, we're putting a valve in. So after my mouth, after my esophagus, the same kind of logic. So this is my lower esophageal or my gastroesophageal. So where do you think the next one would be? It's actually in the animation. Let's find out. Here's my pyloric valve or sphincter. You can say it either way. There's a subtle distinction. But my third one is going to be my pyloric valve or sphincter. And how would you describe that to somebody? Where's it at? Bottom of the stomach. Or you can say the top of the small intestine, or you can say between the small intestine and stomach. I don't care how you say it. We'll do that one. So here's my pyloric. So it's closed. I open the door to let the food out of the stomach, and I close the door again. Make sense? Okay, Smarties. Where do you think the next one will be? Very good. Let's go to this picture. Say hello to my colon. Your small intestine was in the middle. But here it is, right there. There's a valve or sphincter. That's your ileocecal. We usually call it a valve. But you can call it a sphincter for today. So number 4 is my ileocecal valve. And if you put a hyphen in it, that'll help you remember it's between your ileum and your cecum. That's why they named it that. So where is that in your body? Between what and what? Between small and large intestines. So it's between small intestine and large intestine. Or you can say after the small intestine. You can say before the large intestine. But it's between those two. Between my ileum and my cecum. Okay, let's keep going. We only have 2 more to go. Indeed, indeed. Let's play around with your internal sphincters. All right, internal anal sphincter. And this one shows an external anal sphincter. Where's your anal sphincter? I think we all know, right? Find your anus. All right. Here's my question. Why have 2? None of these other ones have 2.
One is controlled and one is uncontrolled.
That is exactly right. So which one is the involuntary sphincter that you do not have control over? Internal is involuntary. That's a pressure release valve. If there's too much poo, it will open. So therefore, external must be what? Voluntary. So when your children become potty trained, you've taught them where their external anal sphincter lives. Now they control it. So let's keep going backwards then. Your ileocecal, is that voluntary or involuntary? Involuntary. Your pyloric sphincter, is that voluntary or involuntary? Involuntary. Your lower esophageal, is that voluntary or involuntary? Involuntary. Your upper esophageal.
Involuntary.
Alright, let me show you a picture again.
Uh-oh.
Go back to this picture that I lost over here. Alright, so the sphincter is actually not the back of the mouth. It's the one by your larynx, right there. So I want you to open and close that for me right now.
You can't really.
You can't really. Now you can voluntarily swallow or not. But you really can't voluntarily open and close the back of your throat. So most people, including myself, will argue that's an involuntary sphincter. And before you raise your hand, what about sword swallowers? You can elude or override that at some point. But it takes a lot of training. But for most people, you don't get to control that. For this class, pretty much we're going to call it involuntary. Make sense? There is some argument about that. Yes?
When you're brushing your teeth and you might hit the back of your mouth, is that opening up your upper esophageal sphincter?
Nope, actually that's you'd have to shove your toothbrush down your throat right here. What you did was you hit the uvula, which immediately makes this contract and gag to prevent something from jamming the sphincter.
Okay.
Uvula is basically
But
a size
if
determinant.
someone's trying to induce vomiting,
Right.
that's
What you're trying to do
your
is
upper
force that
esophageal.
sphincter to open.
Okay.
So you okay in your sphincters? Alright, so let's go back to our case study, which is why you're here and learning this. Let's find out our executive guy. So what normally prevents gastric reflux? It's a sphincter, which one? The lower esophageal. That's the one that's going to stop stuff from my stomach up into my throat. In this case, they call it the LES, the lower esophageal sphincter. Make sense? So he's having a problem with sphincter number 2. Make sense? Let's figure out the question 2 then. Why is his reflux worse after a meal? What about the folds? Yeah, you're right. Folds. What about folds?
Wouldn't it also be contracting?
Yes. Let's look at the picture. So find again, where's my lower esophageal in this picture? Right up here, right? So let's say I have a big meal, and I fill up my stomach all the way to the tippy top with my beautiful meal. What's that going to do to the pressure on my sphincter? It can push more. My stomach starts to churn. What's that food going to do to that sphincter? You're going to push on it, and if it's a weak sphincter, what will happen to all that food? Comes on up. And what's that food floating in? Acids. What does the acid do? Right. So the idea is the fuller my stomach, the closer the food is to the valve. The more pressure you put on the valve, the more pressure you put on the stomach. Okay? So I'm just filling this up enough to keep beating on that sphincter over and over and over. Make sense? Just putting more force on the valving. More food to splash. If you know someone with GERD, they keep telling me to eat small meals. That's the logic behind it. Okay on that one? Mm-hmm. So let's go back to our case. They did this big expensive thing called Esophageal Manometry, which means it measures the pH. So let's go back to your chart of doom. What was the pH of your esophagus normally? Neutral. What's the pH of your stomach normally? So if I measured your esophagus, if you had this GERD, what would I see?
Acidic.
It'd be acidic, right? Because normally an esophagus is neutral. The only way it could be acidic is my stomach's coming up to meet it. Okay? So my esophagus would get more and more acidic from the acid from the stomach. Make sense? It's like a proof to it. So more and more acidic. pH of 4, pH of 3. You okay with that idea? Okay, so I'm going to give you and your team about 30 seconds to do number 4. This is actually a review from 232. For this patient, do you want to give them a cholinergic agonist or an andronergic agonist? I'll give you 30 seconds to be totally clueless about that answer. Just see if you can do it.
One sympathetic.
Let's
So
say
rest and digest would be
No, it would be cholinergic, and this is something that increases. If it was an antagonist, it would slow it down.
Yeah.
So I think you'd want something that increases digestion. Unless the thing.
But rest and digest, you digest more when you're resting and digesting, right?
So, yeah. So you're thinking you would encourage the androgenic response?
Yeah, that's what I
stomach
think.
acid? I can see that.
ACH.
He's trying to remind us of what we studied last
Yeah. Yeah, so we were right? Andrew. Wait.
Okay, your time is up. So let's review for everybody's benefit. If you forgot, I can still change your grade. Cholinergic was referring to acetylcholine, right? Now the primary neurotransmitter is the parasympathetic system. Andronergic was adrenaline, which is norepinephrine, which is primary neurotransmitter of the sympathetic nervous system, right? Okay, so you have to go back to what these systems do to stomachs. Parasympathetic was rest and digest. And what does your rest and digest system do to stomach activity? It increases rest and digest, so it has more stomach. Right. Sympathetic is fight or flight. And what does fight or flighting do to your stomach activity?
It stops
Decreases
it.
your stomach activity, right? So then the next word is this word agonist. What does agonist mean in doctor speak?
Increases?
Increases, same as, encourages, benefits too. So in English, do I want more parasympathetic or more sympathetic? I want more sympathetic because that would shut off his stomach activity. Less acid, less contractions, less everything. So in this case, if I turn on and on, we have turn off his histamine for number 5. Those would both shut off his stomach, thereby reducing the acid and motility. Make sense? Let's pretend he actually wants to do it the old-fashioned way, which is diet. So tell me what this person should and should not be eating if he has GERD.
Acidic food.
Acidic foods wouldn't be good, right? Coffee will kill you.
Huge meal.
Big meal. So let's do that. Why would a big meal matter?
Because it fills the stomach.
Fills the stomach, more pressure on my sphincter, right? So big meals would be bad. What other types of foods would make my stomach be more active? Alcohol, spicy foods, also protein food, right? Because protein's broken down in my stomach. So worthless trivia, when I was a lad, they used to call this the businessman sales lunch problem. right? You wine and dine your clients, you take them for a big steak and eggs meal, you have a cigar, a bunch of coffee, and pretty soon you're throwing up a bunch of acid. So all that stuff makes your stomach more agitated, more full. Make sense? So there's a problem with this sphincter letting the acid out. Make sense? So flip the page over then. We have another case that sounds exactly like the first one. There's a subtle distinction. Okay, here we got burning, gnawing, okay, the same kind of stuff. I'm getting awake from pain. But let's figure out what happened. They found out that's a duodenal ulcer. So we have two words here we're going to figure out. Can someone tell me where duodenal means in English? Duodenum is your small intestine. So duodenal ulcer means a small intestine. Ulcer means what?
A hole.
A hole. So literally you have a hole in the lining of your small intestine. Duodenal ulcer. So tell me, smarties, peptic ulcer. What's the difference there? Stomach hole. Pepsid-AC, Pepto-Bismol, Pepsin. They all mean stomach. So the only difference between these two things is where the hole is located. In a duodenal ulcer, the hole is in your small intestine. In your stomach, it's in your stomach. It's the same kind of concept. Make sense? So tell me, for question 2, how could you get a hole in your small intestine?
Acid.
Acid. Ooh. Well, why would the acid burn a hole in my small intestine? Describe a small intestine to me. It's thin. And what does acid do to thin things?
It burns.
Burns right through. Remember, no Dorito effect. So you're going to have a problem with acid. So the main problem is acid is burning a hole, and then bacteria move in for the kill, begin chewing on you, because now there's a hole in you. Right? H. pylori. So we can argue this person has the same problem as the last page. They have too much acid screwing up their body. Make sense? Okay, now, this question 3 is new. So we got to figure out what in the world that stuff is. So let's go over here and figure out what in the world gastrin is. So we have another chemical called gastrin. What is it, say ye? All right. Green, green, green. Gastrin is a hormone, not an enzyme. So again, classic test question is you start listing it as an enzyme. I scream in red. So remind me from last term what hormones do. What are they for?
Communication.
Communication, right? They go from organ to organ. So last term you learned source and target and action of hormones, right? So, can someone tell me, gastrin, what the source is? The stomach. So your stomach is what makes gastrin, located there. Can someone guess the target of gastrin? The stomach, again. This is the most bizarre creature in the planet. It's like mailing a letter to yourself. Right? You produce it, it goes in the circulatory system, ends up right back where it was.
Is it in a different tissue?
Yeah, slightly different cell. Right? But it's the same organ. The action. Right. It's going to increase stomach activity, which in English means making acid, grawling, churning, all that. So it turns on my stomach. An English way to say it. Turn on stomach. So it turns my stomach on. When do you think it does that? When in your lifetime would you want your stomach to be on? When you eat. So when you eat food, you need to turn on your stomach and tell it to start doing its job. So you're going to produce gastrin that turns on the stomach. So when do you turn off gastrin? When you're not eating. So if you're not eating, what should happen to gastrin levels if you do not eat? It should go down. Because I sure don't want gastrin if I'm not eating something. Right? So let me show you an animation of gastrin. Last one for today. Okay. That. We'll talk more about these phases they mentioned next week when we finish. But for right now, we'll just focus on the gastrin stuff.
Regulation of stomach secretion is divided into 3 phases: cephalic, gastric, and intestinal. In the cephalic phase, the taste or smell of food, tactile sensation of food in the mouth, or thought of food sends nervous impulses to the medulla oblongata.
Wow, why would we use a medulla for this? This sounds kind of silly. What's your medulla for? Is eating life support? Last time I checked, right? So your brainstem is going to be dealing a lot with this, but that's just gee whiz.
These impulses cause parasympathetic neurons via the vagus nerves to stimulate secretion of hydrochloric acid and pepsin in the stomach.
Ooh, why are we using parasympathetic here? Rest and digest, right? I have to have parasympathetic to be resting and digesting. So I'm turning on my stomach, but lo and behold, now we're going to use gastrin.
The parasympathetic stimulation also results in secretion of gastrin from the lower part of the stomach.
Remember, this is not an enzyme. It's not going to digest anything. It just turns on my stomach. So it's going to go in my bloodstream and end up right back basically where it started.
This hormone travels through the bloodstream and further stimulates hydrochloric acid and pepsin secretion in the upper and middle parts of the stomach.
It's basically 2 doors down.
In the gastric phase of gastric secretion, food has entered and distended the stomach.
Right, so what we're seeing is, as now I'm eating, my gastrin levels should keep climbing because I'm going to turn on my stomach. Make sense? Okay, so we'll pause the rest of that until next time. But let's look back at our case. So look at question 3. They did a pentagastrin stimulation test. For future nurses, pentagastrin means, right here, a gastrin agonist. What does agonist mean again?
Increase.
Increases, same as encourages development. Okay, so in English, I'm giving you more gastrin, is what that phrase means. So let's look what happened. Demonstrated the gastric juice pH fell to 1.9. What does that mean in English if pH falls? Okay, is that normal for the stomach to get more acidic if I give a gastrin?
Yes.
Yes, indeed. So the doctor's proving that your stomach is responding to gastrin. So I give you gastrin, you should turn on a stomach, which means makes acid, right? Get more acidic. Make sense? Okay, look what happened in the fasting level. Fasting means I didn't eat. Tell me what the gastrin level was when I stopped eating. Normal near what?
0.
Is that normal? Yes, indeed. Because if I'm not eating, gastrin levels should go down. So what the doctor's trying to do is prove if your stomach is making too much acid. And from the data, what can we conclude? Perfectly fine. Your stomach is turning on when it should, it's turning off when it should. So it's not your stomach's problem that you have this ulcer. Makes sense the logic there? At least for this case study. Right? So I turn on a stomach with gastrin, I turn it off when you're fasting, your stomach is fine. So therefore we got to figure out why they have this ulcer if the stomach isn't the cause. So we got to go down to question 5. Trick is why the pain goes away when they eat, but then is there later in the day. So there must be something else going on. So look at your chart of doom again. Find the pH column. What was the pH of your stomach?
Basic. Oh, acidic.
Acidic. What was the pH of your intestine?
Basic.
How in the world did you go from pH of 2 to pH of 8 in 1 inch? How do you change the pH of a small intestine?
A leak.
So I have a job. Let's talk about buffers for a little bit. Buffer. There's one more chemical we have to add to your chart. And that is this one here. Pancreatic buffers. Biology 112. Can you remind me what buffers did?
It helps things stabilize.
That's right. Stops acidity or stabilizes pH, right? How do you want to phrase it? So where are these buffers made? pancreas. Pancreas puts them into what organ? Into the small intestine. Okay? So my pancreas's job, one of them, is to make a buffer to put in my small intestine. And why would the small intestine want to stop acid? Because it's thin and it'll get destroyed by your stomach. So the stomach dumps acid in the small intestine has to stop itself from getting destroyed. Right? That's because the pancreas makes a buffer. Right? So let's go back to question 5. So, when you first eat a meal, what are you going to do with the buffer that's in your pancreas? You're going to release it where? The small intestine. And now let's say you run out of buffer a little bit later, and you're still eating. What will happen to that acid if you no longer have enough buffer? It's going to burn. What will your stomach, your small intestine do if you don't have enough buffer to compensate the acid? It will get an ulcer. So what we're proving in this case study is the stomach's doing its job fine. You don't have the buffers to stop the acidity. And the buffer, the main one, is called bicarbonate. Hey, where have you heard about bicarbonate before last week? Right? That was the HCO3.
In your blood.
So one of the jobs of your pancreas is to add HCO3 to your small intestine so your small intestine doesn't burn and go away. If you run out of buffer, you'll start getting pain when the buffer runs out, which is step number 5. So they're running out of buffer to neutralize the stomach's acidity. So then let's go back to question 6 then. Give me a treatment. So if you don't have enough buffer, what can you do? Eating comes and acids, right? That's the logic, is this stops acidity. What could you do dietarily to maybe help this?
Less citrus.
Less acidic foods. Ice cream does help. Right? That's fine. Smaller meals, all that kind of stuff. The same exact answer you're going to give me for the guy on the first page. So the guy on the first page, the problem was he had too much acid up his throat from a sphincter problem. The person on the second page has too much acid in the duodenum from a buffer problem. They both have too much acid in their system. Right? Makes sense? Yes?
Would you ever see
Yes. The caloric valve is staying open, which is, it's a fancy word for it, escaping it. Yeah, but it doesn't close. Then you get acid all the time leaking in, and that would also do that. As long as the stomach is shutting off, it should stop. Make sense? So one other thing your pancreas does is buffers. Make sense? Question. Thanks, yes.
So what's if the solution is fine, is there a this solution that are using buffer and you're not using?
They just run out of the buffer. So what happens is the food takes about an hour to get through.
Okay.
So you use up the buffer initially, the food's still trickling in from the stomach, and so it hits and there's no buffer to neutralize it. So then it just sits there and burns. So smaller meals.
Is that just a deficiency of the pancreas then?
It can be. Or pancreatic cancer, or too big of a meal. Make sense on these? Yes?
If you continue to eat spicy food, would you increase your buffer production?
Good question. I don't know. I want to say no, but I don't have anything other than my gut.
Oh, like for cultures that
Yes.
tend to eat spicier food.
I've never heard anyone say that. I don't know. I'm going to plead ignorance now.
Can you eat?
Dr. Google would know. Questions? All right. Let's do one more little thing here. So let's go back to that video we were seeing. It was right here. We're going to add a little more to our stomach here. So here was our stomach. Remember, we saw the food, we made the gastrin.
This distension activates a parasympathetic reflex via the medulla oblongata and also has a direct stimulatory effect on the gastric glands. The result is continued secretion of hydrochloric acid and pepsin.
As long as I'm eating the meal, I should keep making on the gastrin, turning on my stomach. But what we're going to do is shift slightly to that HCl stuff. So I'm going to show you this video. And this is more to understand when we do acid-base balance in about 5 weeks. This video will haunt you. So I'm going to show it to you now so you just get the gist of what we're talking about. But here's a stomach cell. So your Whopper with cheese on the right. This is some bloody stuff on the left. How in the world do you make hydrochloric acid in your body? Where's it come from?
Carbon dioxide?
Indeed it is. So So let's figure that out.
Hydrochloric acid is produced in parietal cells through a complex series of reactions. Carbon dioxide diffuses.
CO2. All right, so remind me from last week, what does CO2 make in your body? It makes what? That's the same logic. So no matter what acid we're talking about, you're essentially going to have CO2 as the base product that you use. All right, so every acid is pretty much going to use CO2 from your tissues. And you're going to convert it into HCl. So let's do that.
Into the parietal cell, and the enzyme carbonic anhydrase catalyzes a reaction between the carbon dioxide and water.
That's CO2 and that's water. You know what the next part is on the equation from last week. CO2 plus water makes what? Okay, H2CO3.
Water to form carbonic acid.
Not that.
Carbonic acid dissociates into bicarbonate ion and hydrogen ion.
Right there. So I split that. Here's that H plus and HCO3 minus. Okay, now if you look at what I just wrote over there for the pancreas, what's the main buffer of the pancreas? Bicarbonate. Hey, I just made bicarbonate. Where should I send that? My pancreas. So waste not, want not. So I just made an acid and a base. I'm going to take the base, give it to my pancreas to use for next time, and keep the acid in my stomach. So all the reactions we're talking about are basically the same reaction. All you're doing is separating the products in different body parts. So my stomach's going to keep the acid part and give the pancreas the base part. Cool, huh? Here we go.
And the bicarbonate ion is transported back into the bloodstream.
Go visit Mr. Pancreas.
An ion exchange molecule in the plasma membrane exchanges bicarbonate going out for chloride coming in.
Okay, Saladin goes on and on about the chloride shift. You ever read that? Do we need to read him? Okay, no. But anyway, he goes on about the chloride shift, and that's what they're showing there. I took something out and brought chlorine in. Why in the world do that?
Is it basic?
That's much basic. Okay, what was the charge of my bicarbonate? Let's find out. Negative. So if I take negative out of the cell and don't replace it, what will my cell's charge eventually become? Eventually become positive, right? You learn that with neurons. You can't just change charges of cells very well. So if I want to keep the cell essentially neutral, I have to swap the charges. So I'm going to take the negative HCO3 out and bring in a negative Cl in. That keeps my charge still neutral, right? But hey, that's chlorine. How? Where have I heard chloride before? Bingo. So I'm going to use that step.
Bicarbonate going out for chloride coming in. The hydrogen ions are actively transported into the duct of the gastric gland, and the negatively charged chloride ions diffuse with the positively charged hydrogen ions. Potassium ions are counter-transported into the parietal cell in exchange for hydrogen ions.
Swap there.
The net result is production of hydrochloric acid in the parietal cells and its secretion into the duct of the gastric gland.
So same reaction you learned last week, you're going to see again and again and again. Anytime you're dealing with acids and bases in the body, it's the same reaction. Nothing has really changed. So when we get to acid-base balance, you want to keep this in your head that what I throw up and what I'm breathing are the same chemical. It's CO2 either way. Just all you're doing is changing the form and what cell is using it. Yes?
Is the chlorine coming just from like dissociation of salt?
Yes.
Stuff like that? Okay.
So you're getting Cl, you just dissociate it, and eventually get chlorine to float around. Make sense? So that will come back on us about 5 weeks now. So don't learn that for today, other than that you're making acid. But when we do acid-base balance, that's an important concept to keep in mind. It's the same equation. We're just using it in different forms, different bodies. Make sense? Questions on the chart of doom? Okay, your quiz next week will be on that chart. Be able to reproduce it ad nauseum. I'll let you go early tonight. How's that? You have project groups to pick by next week. Give you time to fill it in, but first we're going to figure out what it's talking about and what the words mean. So on the left-hand side, we have organs that my cheeseburger would flow through. My mouth, my esophagus, down to my butt. Okay? We have columns with yeses and nos. So let's do the first one. Physical digestion. You said, what does it mean to physically digest my cheeseburgers? Chewing, ripping, tearing, stomping, grinding. You're physically making the pieces get smaller. Let's do this together. Does your mouth physically digest?
Yes.
Yes. So you're going to put a yes there. Next column says chemical digestion. What does that mean in English to chemically digest? Enzymes, you're melting, your hydrolysis, whatever word you like. You're melting the food, dissolving the food using enzymatic hydrolysis, whatever word you like. So, does your mouth chemically digest? Yes, that's called spit, right? That's why you have it. So I physically and chemically digest my cheeseburger. I have teeth and I have spit. Now, last column with yeses and nos. Nutrient absorption. What does that mean in English? Absorption? Got that. What does that mean in English, though? Absorb them where? Into your bloodstream. Very good. So I'm taking the food out of my organ and putting it into my bloodstream. Does your mouth absorb cheeseburgers? No. But Mr. Christie, what about nitroglycerin? We're not talking about pharmaceuticals. We're not talking about under the tongue, sugar pills. We're talking about, like, food. Put a cheeseburger under your tongue. It's still there. I've tried this. I have a friend. Sorry. All right, so food we do not absorb in your mouth. Right? So we're talking about food. So you're going to have a yes, yes, and a no. When you get to the pH column, I don't need a number. I just need a value. Is it acidic, neutral, or basic? Okay? I'm not going to tell you that one. When you get to enzymes present, that's not a yes or no. I need the names of the enzymes in the mouth. Your lab manual has a sheet, so does your book, and so does your phone. So you can do that. Anyone know what enzyme source means from Biology 112? Where they're made or where they're produced. So they're in my mouth, but where are they made? This wording comes from 112, that class none of you should have passed. Right. Let's review Biology 112. I think it's the fourth lab of the term. You had something look like this. You had an E, which was an enzyme. You had a little triangle that went into it. Right? You drew something like this. And then the next week you did something kind of like this. Right? The S represented substrate. So in English, that's what the enzyme works on. So substrate is what you started with. What'd you call the two little things that you made over here? Products is what you made. So for each enzyme that you list, you're going to list what goes in, what comes out. Substrates and the product for each enzyme. Make sense? Okay, your job, I'm going to give you about 10-15 minutes with your team, divide and conquer, but fill that in, we'll go over it. Have fun.